Good day, this is Professor Will, CBMD PhD. Today is Wednesday, April 29th, 2020. It is 6:49 and 12 seconds a.m. Eastern Standard Time. And this is a continuation of my COVID-19 corona research. So let's get started here. Key node cells identified as likely COVID-19 virus entry points. Human Cell Atlas study could help understand transmission of the virus. Date April 23rd, 2020. Source Wellcome Trust Sanger Institute. Summary. Two specific node cell types have been identified as likely initial injection points for COVID-19 coronavirus. Scientists discovered that goblet and ciliated cells in the nose have high levels of the entry proteins that the COVID-19 virus uses to get into our cells, which could help explain the high rate of transmission. The study with Human Cell Atlas, a long biological network, found cells in the eye and some other organs also contain the viral entry protein. Reported today, 23rd April, in Nature Medicine. This first publication with the Lung Biological Network is part of the ongoing international effort to use Human Cell Atlas data to understand infection and disease. It further shows that cells in the eye and some other organs also contain the viral entry protein. The study also predicts how a key entry protein is regulated with other immune system genes, and reveals potential targets for the development of treatments to reduce transmission. Novel coronavirus disease, COVID-19, affects the lungs and airways. Patient symptoms can be flu-like, including fever, coughing, and sore throat, while some people may not experience symptoms but still have transmissible virus. The virus is thought to be spread through respiratory droplets produced when an infected person coughs or sneezes, and appears to be easily transmitted within affected areas. So far, the virus has spread to more than 184 countries and claimed more than 180,000 lives. Scientists around the world are trying to understand exactly how the virus spreads, to help prevent transmission and develop a vaccine. While it is known that the virus that causes COVID-19 disease, known as SARS-CoV-2, uses a similar mechanism to infect our cells as a related coronavirus that caused the 2003 SARS epidemic, the exact cell types involved in the nose had not previously been pinpointed. To discover which cells could be involved in COVID-19 transmission, researchers analyzed multiple Human Cell Atlas (HCA) consortium datasets of single-cell RNA sequencing from more than 20 different tissues of non-infected people. These included cells from the lung, nasal cavity, eye, gut, heart, kidney, and liver. The researchers looked for which individual cells express both of two key entry proteins that are used by the COVID-19 virus to infect our cells. We found that the receptor protein ACE2 and TMPRSS2 protease that can activate SARS-CoV-2 entry are expressed in cells in different organs, including the cells on the inner lining of the nose. We then revealed that mucus-producing goblet cells and ciliated cells in the nose had the highest levels of both these COVID-19 virus proteins of all cells in the airway. This makes these cells the most likely initial infected route for the virus. This is the first time these particular cells in the nose have been associated with COVID-19. While there are many factors that contribute to the virus transmissibility, our findings are consistent with the rapid infection rates of the virus seen so far. The location of these cells on the surface of the inside of the nose make them highly accessible to the virus, and also may assist with transmission to other people. The two key entry proteins, ACE2 and TMPRSS2, were also found in the cells in the cornea of the eye and in the lining of the intestine. This suggests another possible route of infection via the eye and tear ducts, and also revealed the potential for fecal-oral transmission. When cells are damaged or fighting an infection, various immune genes are activated. The study showed that ACE2 receptor production in those cells is probably switched on at the same time as these other immune genes. The work was carried out as part of the Global Human Cell Atlas consortium, which aims to create reference maps of all human cells to understand health and disease. More than 1,600 people across 70 countries are involved in the HCA community. The data is openly available to scientists worldwide. By pinpointing the exact characteristics of every single cell type, the Human Cell Atlas is helping scientists to diagnose, monitor, and treat disease including COVID-19 in a completely new way. Researchers around the world are working at an unprecedented pace to deepen our understanding of COVID-19, and this new research is testament to this. Collaborating across borders and openly sharing research is crucial to developing effective diagnosis, treatments, and vaccines quickly. ensuring no country is left behind. Researchers identify cells likely targeted by COVID-19 virus. Study finds specific cells in the lungs, nasal passages, and intestines that are more susceptible to infection. Date April 22, 2020. Source Massachusetts Institute of Technology. Summary. Researchers have identified specific types of cells that appear to be the targets of the SARS-CoV-2 coronavirus that is causing the COVID-19 pandemic. Using existing data on the RNA found in different types of cells, the researchers were able to search for cells that express the two proteins that help the SARS-CoV-2 virus enter human cells. They found subsets of cells in the lung, the nasal passages, and in the intestine that express RNA for both of these proteins much more than other cells. The researchers hope that their findings will help guide scientists who are working on developing new drug treatments or testing existing drugs that could be repurposed for treating COVID-19. Our goal is to get information out to the community and to share data as soon as humanly possible, so that we can help accelerate ongoing efforts in the scientific and medical community. Digging into data. Not long after the SARS-CoV-2 outbreak began, scientists discovered that the viral spike protein binds to a receptor on human cells known as angiotensin-converting enzyme 2, ACE2. Another human protein, an enzyme called TMPRSS2, helps to activate the coronavirus spike protein to allow for cell entry. The combined binding and activation allows the virus to get into host cells. As soon as we realized that the role of these proteins had been biochemically confirmed, we started looking to see where those genes were in our existing data sets. We were really in a good position to start to investigate which are the cells that this virus might actually target. Many other labs around the world have performed large-scale studies of tens of thousands of human, non-human primate and mouse cells, in which they use single-cell RNA sequencing technology to determine which genes are turned on in a given cell type. Researchers have been building a database with partners at the Broad Institute to store a huge collection of these data sets in one place, allowing researchers to study potential roles for particular cells in a variety of infectious disease. Much of the data came from labs that belong to the Human Cell Atlas project, whose goal is to catalog the distinctive patterns of gene activity for every cell type in the human body. The data sets that the MIT team used for this study included hundreds of cell types from the lungs, nasal passages, and intestine. The researchers chose those organs in the COVID-19 study because previous evidence had indicated that the virus can infect each of them. They then compared their results to cell types from uninfected organs. Because we have this incredible repository of information, we were able to begin to look at what would be likely target cells for infection. Even though these data sets weren't designed specifically to study COVID, it has hopefully given us a jump start on identifying some of these things that might be relevant there. In the nasal passages, the researchers found that globulate secretory cells which produce mucus-expressed RNAs for both the proteins that SARS-CoV-2 uses to infect cells. In the lungs, they found the RNAs for these proteins mainly in cells called type 2 pneumonocytes. These cell lines the alveoli, air sacs of the lungs, and are responsible for keeping them open. In the intestine, they found that cells called absorptive enterocytes, which are responsible for the absorption of some nutrients, express the RNAs for these two proteins more than any other intestinal cell type. This may not be the full story, but it definitely paints a much more precise picture than where the field stood before. Now we can say, with some level of confidence, that these receptors are expressed on these specific cells and these tissues. In their data, the researchers also saw a surprising phenomenon. Expression of the ACE2, a gene, appeared to be correlated with activation of genes that are known to be turned on by interferon, a protein that the body produces in response to viral infection. To explore this further, the researchers performed new experiments in which they treated cells that line the airway with interferon, and they discovered that the treatment did indeed turn on the ACE2 gene. Interferon helps to fight off infection by interfering with viral replication and helping to activate immune cells. It also turns on a distinctive set of genes that help cells fight off infection. Previous studies have suggested that ACE2 play a role in helping lung cells to tolerate damage, but this is the first time that ACE2 has been connected with the interferon response. The findings suggest that coronaviruses may have evolved to take advantage of the host cell's natural defenses, hijacking some proteins for their own use. There are other examples of coronaviruses and other viruses that actually target interferon-stimulated genes as ways of getting into cells. In a way, it is the most reliable response to the host. Because interferon has so many beneficial effects against viral infection, it is sometimes used to treat infections such as hepatitis B and hepatitis C. The findings of the MIT team suggest that interferon's potential role in fighting COVID-19 may be complex. On one hand, it can stimulate genes that fight off infection or help cells survive damage, but on the other hand, it may provide extra targets that help the virus infect more cells. It is hard to make any broad conclusions about the role of interferon against the virus. The only way that we'll begin to understand that is through carefully controlled clinical trials. What we are trying to do is put information out there because there are so many rapid clinical responses that people are making. We are trying to make them aware of things that might be relevant. Researchers hope to work with collaborators to profile tissue models that incorporate the cells identified in the study. Such models could be used to test existing antiviral drugs and predict how they might affect SARS-CoV-2 infection. The MIT team and their collaborators have made all the data they used in this study available to other labs who want to use it. Much of the data used in this study was generated in collaboration with researchers around the world who were willing to share it. There has been an incredible outpouring of information from the scientific community, with a number of different parties interested in contributing to the battle against COVID in any way possible. It is incredible to see a large number of labs from around the world come together to try and collaborate tackle this. Good day, this is Professor Will, CBMD PhD on microbiology and pathophysiology. Today I'd like to make a very special announcement. This is my 35th anniversary of studying encyclopedia mathematics and much more. Well, this concludes today's reading on Corona COVID-19. Hope you enjoyed listening and have a great day.